Okay, so start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you for joining us the 42nd seminar, and I call it Purdue University Day. This is the rescheduled seminar due to some difficulty scheduling last time. For me, this week is a rare week without a seminar trip this summer. In fact, I'll be giving more than 50 seminar or conference presentation this year again, but mostly in person compared to last year. It is, however, great to spend more time with my family this week. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Mike Ladisch. He is a distinguished professor at Purdue University. He does not need any introduction because we all know him. But in short, he is a member of National Academy of Engineering. And what else do you want to know about him? So Professor Ladisch, the virtual podium is all yours, and thank you so much for your time out of your busy schedule. Thank you.
Well, thank you very much, and it's really a pleasure to be included with a list of long distinguished young speakers and exciting research topics. And as you will introduce in a few moments, Alexander will be presenting his work on CRISPR and the dynamic regulation of enzymes, and has a very clear and compelling presentation. I've had the pleasure of looking at both the PowerPoints, and thank you very much also, Alexander, for sending your Nature paper. And for those of us in the field of CRISPR, I would highly recommend reading it. It's really very clearly written, particularly for more traditional chemical engineers myself, and really is quite fascinating. One thing I'd like to mention, and we were just having a sidebar before we started, is that CRISPR actually has evolved from the food industry and the food field. And basically, there was a researcher at DuPont, now at North Carolina State University, who had discovered that certain batches of yogurt will go bad. And the reason is that there would be a phage infection. And in those batches where the yogurt would no longer be suitable for consumption, it turned out the microorganisms involved would be attacked by the phage. Yet some batches had the same phage and were fine. And this led to the discovery of CRISPR, which is basically a bacterial immune system. And this is since repurposed in many, many ways that we have discovered along the way, and basically something that Alexander will introduce us to when he gives us the presentation. But overall, Tae-Sok asked me for some comments, and I think for all of the young bioengineers and scientists, bioprocess scientists, this is a very exciting time with the challenges of food security, climate change, okay, and greenhouse gases, certain advances in medicine, and the prevalence of viral infectious disease. We've just been through this. There are many opportunities to use our craft, our knowledge, our education to make the world a better place. And it starts in the laboratory. It transcends any one university or company, I believe, or country, as a matter of fact, and rather basically involves education of future generations of students who will address these areas, which are very complex, require multiple disciplines, both engineering, social, and science, because in some cases one may have a solution, but if it's not accepted by society or the people that make up our society, it's not a very good solution either. And the last comment I'd like to leave you with is engineering is important, because we have to look towards translating our discoveries, technologies, and advances into actual practice. It's very important to have publications so that we communicate our results to our peers and others, but it's very, very important for the combination of our knowledge to be put into practice. And I think, again, Alex will describe some examples of that. And so the translation component, which means scaling up, manufacturing in an environmentally acceptable way, then being able to distribute the product, provide a product which is stable over time, and can be equally used, whether it's in a developed country or in countries where, or areas of any country where perhaps the people are not as fortunate. And so, for example, some areas in the world do not have a lot of refrigeration, just as an example, or storage of food, etc. So all these factors must be considered as well. So with this, I think I would end my comments and just encourage this impressive series to continue. And certainly, in a way, TASOC is trying to catalyze networking, I should say. And so again, thank you very much, and I'll turn it over to TASOC and to Alex.
Thank you so much. That is every single point you made basically touched me so much, because that's the all important thing, especially about education. That is brilliant thing, because we have too many problems in the world right now, including food security, climate crisis, medicine, and also many other things. But I believe, because of young people we have, our future is not gloomy but bright, because we rely on them. They are doing fantastic job, industry, academia, and then I believe we will solve many problems together through collaboration. And then your vision, will be implemented by them. So I'm super excited. And as you mentioned, I will continue this seminar series until I die, forever, as long as they support me. So thank you so much. I mean, this is brilliant. I love it. Thank you. Thank you. Okay, so now the main speaker of today with the longer introduction. So Alex Mitkas is now at Pfizer as a senior scientist and received his PhD degree at University of Delaware. He was working with Professor Winfred Chen, and actually I love him a lot, and his work is brilliant. And with a thesis focusing on developing a dynamic scaffold system for the in vivo co-localization of proteins using CRISPR-Cas6 proteins and RNA-RNA interaction. I believe he will talk about this one today. The platform has been developed to demonstrate control of metabolic flux and has been successfully applied to increase the titer of non-native products. And this is probably aligned with what Mike just mentioned about real application, because many of us is engineer, and that application is very important. Before joining University of Delaware, he completed his undergraduate degree at MIT, and his research focused on combinatorial immunotherapies to combat skin cancer, another important area of research. So Alexander, thanks so much, and take it away, and that's all yours now. Thank you.
Thank you for that introduction, both. You can see my screen, yes?
Yes. Slide mode, please.
Okay, yep.
Okay, wonderful.
Well, thank you everyone for joining me. Today I'll be talking to you about my thesis work at University of Delaware on developing a high binding affinity dynamic modular scaffold toolkit for the control of intracellular metabolic flux. Before I get into the actual nitty-gritty, I just wanted to give a brief overview of what I'll be talking to you about. First, I'll give some background and motivation. Follow that, I'll describe how we propose to design our system. Then the biggest part will be the experimental work we did to validate and demonstrate the functionality of the system. And finally, I'll close with some conclusion and future directions. So our motivation for this stems from humanity using cells as a way to produce complex chemicals since the beginning of time. It started with people discovering that you can ferment milk and grape juice to make cheese and wine. And then as science evolved, so did our understanding of these tiny organisms, and people started to isolate more specialized organisms such as Clostridium acetobutylicum for acetone, butanol, and ethanol synthesis, as well as P. chrysogenum for penicillin production. And that has culminated to where we are right now, where we have modern metabolic engineering, where we try to actually harness the cell's productive power and use them to make even more hard to make chemicals in greener ways. And the way we do that in metabolic engineering is we like to treat cells as mini factories. So just as a factory will have a feed, waste, and then product, the same way cells can be engineered in order to be treated as factories. So you can engineer them to utilize different substrates, and you can also add different pathways to engineer them to produce chemicals that they would not natively produce. However, there is one big difference between actual factories and cells as factories, and that is that factories are optimized bottom up. So you start building a factory with the goal to produce a few products. Whereas in engineering cells, we follow a top-down approach where we actually try to hijack the cell's resources and machinery for our own goals. So in the factory metaphor, that would be us moving into a different factory and trying to use that to make our own product. The way we do that is we introduce novel metabolic pathways in platform microorganisms such as yeast or E. coli. And then basically a big problem that arises from that is that these strains can often exhibit poor product titer because exactly they're not optimized to make these products. So in order to get around that, we have a few ways to optimize product yield. First one being pathway engineering, where you can figure out where your bottlenecks lie and if your product is being consumed elsewhere as well. And then doing that, you get to get around that, you can either, you can overexpress enzymes that are in a bottleneck pathway, or you can delete enzymes that are consuming your product. Alternatively, what you can do is use directed evolution as a way to create more efficient enzymes or more efficient machines to basically increase their turnover rate and give you higher yields of your product. One kind of problem that is being addressed is being solved in parallel as my proposed approach, but basically you require multiple iterations to get any results of these, and oftentimes these approaches are not generalizable. So what worked for one pathway may not necessarily work for another. And again, there is no guaranteed outcome. So in lieu of all these comments, we proposed to basically use synthetic metabolons as an alternative, which can be made to be modular and are also inspired by nature. In a factory analogy, this would be akin to creating an assembly line with all your enzymes clustered closely together. So metabolons in nature are temporary spatial organizations of enzymes, which increase turnover rate through something called substrate channeling. So when the enzymes are far apart, the intermediates are more likely to diffuse out. However, if you cluster them together, then you can have a higher throughput through that path. And this is especially important in branched pathways where an intermediate can be consumed by one or more than one enzyme. People have already been inspired by this, and scientists have demonstrated that you can create synthetic metabolons to increase yield of products. However, there's one big difference between the synthetic metabolons developed so far and those found in nature, and that is that the synthetic ones are static. So once you turn them on, that's pretty much it. So why would we want a dynamic synthetic metabolon? Well, in chemical engineering processes, we have valves, splitters, and mixers to basically create dynamic control of flow rates and allow us to regulate our process. Similarly, in native cellular processes, we have cell has feedback loops that can help it dynamically regulate flux through different pathways, depending on stimuli it receives from its environment or even from inside its own self. So for engineered processes, that doesn't really exist yet, or not to that extent. So if you add a new pathway to a cell, then you might end up with either adding too much of your substrate pool to an engineered pathway or too little. So if we can find a way to switch between these two, we can kind of help the cell stay healthier longer and be productive for longer, while also getting more of our product in the end. So now that we've established why we need a dynamic metabolon, the next question is, how do we make that? And we want to make a dynamic synthetic metabolon by using RNA interactions for its assembly and disassembly, and specifically RNA strand hybridization for metabolon assembly, and then toehold mediated strand displacement technology for its disassembly. For those of you who are not familiar with toehold mediated strand displacement technology, it is a thermodynamically driven reaction between 3 strands of RNA or DNA. And this begins by hybridization of the trigger strand to the strand B at this 2,2 star region. Once that happens, the trigger strand will start displacing the strand A until it finally has completely displaced it. Now this reaction will pretty much readily happen if the strands are mixed, and essentially its time scale is super fast. It can happen in minutes to hours, and even more importantly, we can tune the kinetics by changing the length of the toehold and by adding mismatches in the toehold. So we can fine tune how fast this reaction happens. And in fact, nowadays there's even computational tools to allow you to predict how fast it will happen. So now that we've established that we want RNA-RNA interactions to be the drivers of the dynamicness of our system, the next question is, how do we ensure that that is the only way our system assembles or disassembles? And to do that, we need a way to scaffold our proteins of interest on RNA with high specificity and affinity. And to do that, we want to use these CRISPR-Cas type 1, Cas6 proteins. Overall, CRISPR is the immune system of the prokaryotic cells, and it is broadly split into 3 types depending on how what proteins are used to carry out these processes. Now, Cas9 is probably the most famous one, with where everything is done by one big protein. However, that protein is a bit too big for our uses. So instead, we looked at this type 1 CRISPR-Cas system, and specifically this small subunit of the system called Cas6 protein. Cas6 proteins are small endoribonucleases that can bind short RNA motifs with very high affinity, with dissociation constants as low as 50 picomolar. And then once they bind that motif, they cleave at the 3' end of it. So their original function is to basically translate the pre-CRISPR RNA into CRISPR RNA. They are proven fusion partners, and even more interesting for us, there exist orthogonal Cas6 proteins from different organisms, which each one has its own unique binding site. So you can imagine if we had a long RNA strand with 3 different Cas6 binding motifs and the corresponding Cas6 proteins, if we mix everything together, each one would bind to its unique hairpin and then cleave at the 3' end, generating these short RNA sequences with a tightly bound protein to it. And that is exactly what the building block of our proposed scaffold system is. So the way we envision this working inside the cell is basically you have your two enzymes of interest fused onto two orthogonal Cas6 proteins, and then you express one scaffold RNA. The Cas6 proteins can then bind to their respective RNA hairpins, cleave at the 3' end, and generate these scaffold building blocks. Following that, if you design these 5' overhangs to be complementary, then you can drive assembly of the scaffold and localization of the enzymes. So just a linguistic note, from now on, I'll be referring to the system as a scaffold, which is just one approach for metabolon formation, whereas metabolons are just the broader term for enzyme assembly. So moving on to the experimental work. The experimental work is broadly broken down into 3 pieces. The first one is demonstrating that our scaffold can assemble with high affinity and specificity. The second one is showing that we can dynamically assemble or disassemble the scaffold with RNA and small molecule cues, and also cycling between on and off states. And finally, the third part is demonstrating that we actually can control metabolic flux in both native and non-native pathways. So on to part 1. To assess the level of assembly of our scaffold, we decided to use the split luciferase reporter system, which is composed of the small bit over here and the large bit shown in blue. The 2 pieces of parts show minimal activity. However, when brought in proximity, they can catalyze the reaction shown to the right and generate luminescent output. We like the system because it has a high signal to noise ratio. So what we did is, first we want to test everything in vitro. And to do that, we expressed these CSY4 large bit and CSY4 small bit fusions and purified them, and then transcribed 3 different RNA molecules in vitro. Molecules A and B were designed to have complementary 5' overhangs to each other, and molecule C was designed as a negative control, which should not be able to hybridize with either of the 2. So in this experiment, we just simply mixed everything together in different combinations to see how the system would behave when we have great control of what goes into the system. And what we saw is that the 2 fusion proteins on their own displayed minimal activity. And then if you mix the 2 proteins without any RNA, there's a small uptick in the activity, and that is due to non-specific interaction between the 2. And finally, when you actually mix the 2 fusion proteins along with the 2 correct RNA sequences, what you see is a 3-fold increase in luminescence, indicating that our system needs all the scaffold components in order for it to assemble. And even more importantly, if you swap out one of the RNA molecules for the negative control one, you see that the luminescence basically drops down back to background, indicating that not only do we need all the components, the RNA and the fusion proteins, but we also need the correct RNA sequence to drive this assembly. The next question is, how does the system perform in vivo? So we moved everything onto plasmids and controlled everything with different inducible promoters, and ran the same experiment in vivo. Except this time we expressed 1 long scaffold RNA instead of the 2 separate ones. And we also replaced one of the CSY4s with the orthogonal CSC3. Again, here, you see that the 2 proteins on their own have very minimal activity. And then if you express just the 2 proteins without any RNA, there is again a small uptick due to that non-specific interaction. However, in this case, when you express all 3 components of the scaffold, we see an even better increase in luminescence, a 9-fold increase in luminescence, indicating that the system behaves even better in vivo. And that could possibly be to just better conservation of the RNA scaffold in vivo. We also confirmed that we are expressing full length of these proteins by running a Western blot for the his tags on the protein. And basically that kind of confirmed that we can drive this assembly of enzymes in vivo. The next question is, how stable is this scaffold? And to do this, we basically ran the same experiment, but over a longer time period and taking time points at 2.5, 5, and 8 hours. On this axis, we have luminescence fold increase, which is calculated as luminescence of a sample where we express both protein and scaffold RNA over the luminescence of a sample where we just express the proteins. We see that while we start at a 9-fold increase down here at 2.5 hours post-induction, at 5 and 8, we see a drastic increase in luminescence, peaking at around 220-fold luminescence. So that indicates to us that we have a stable scaffold assembly. In fact, it's probably stable enough that it begins to accumulate in the cells, which is kind of what drives this drastic increase in luminescence. So now that we kind of demonstrated that we can get everything to assemble both in vitro and in vivo with the specificity we want, the next question is, can we make the scaffold dynamic? To make it dynamic, what we want to do is basically have the same assembled system and be able to express a trigger strand that through toehold mediated strand displacement will drive the two enzymes apart and lead to a reduction in luminescence or eventually throughput through the pathway. So to test this, we had the same system, and what we added is 3 different constructs for the trigger. So the scaffold RNA was expressed as is, and then we combined this with either a fully scrambled trigger, a trigger with a scrambled toehold, or a regular trigger. And out of all these 3, only the regular trigger should be able to drive this assembly. So shown here are the results of that experiment, where on the y-axis, we again have fold increase in luminescence, where this is calculated as the ratio of a sample where you express all these components versus a sample where you only express the proteins in the scaffold RNA. So a ratio of less than 1 is what indicates this assembly for us. So equals 0 is when we induce the trigger, and you see that all these samples are around 1. And then 5 hours post-trigger induction, you see that for both the scrambled trigger and the scrambled toehold trigger, we see no reduction in luminescence due to the induction of the trigger. However, with the regular trigger, we see about a 60% reduction in luminescence 5 hours post-induction. So this tells us that we can have that scaffold disassembly, and it is specifically driven by toehold mediated strand displacement, because even if you scramble the toehold, you are not able to get any reduction. So that shows us that the system is kind of behaving in the way that we proposed. The next question is, can we cycle between on and off states? So to do this, we needed to extend our trigger sequence to include a second toehold. So we start from an on situation, and then by expressing this T1 trigger, we can induce this assembly of the scaffold. And then if we can express a second trigger, T2, what we end up with is basically the system going back to its original configuration. So for this experiment, again, we have the luminescence ratio calculated as protein plus scaffold plus trigger RNA luminescence over protein plus scaffold RNA luminescence. So to do this, we exchange media after each induction to kind of minimize how much of our secondary trigger is being expressed. So our initial ratio is around 1, and then at that point, we induce T1 trigger. And what we see is 2 hours later, luminescence drops 30%. At that point, we perform the media exchange and then induce the second trigger. And we saw that 2 hours after that, we were able to return to control levels, indicating that we are able to cycle between these on and off states. The reduction isn't as prominent, but that could very possibly be improved by engineering some better toehold sequences using these computational methods I mentioned previously. So we have shown that we can go from an on configuration or assembled configuration to an off configuration. The next question is, can we do the opposite? So in this case, what we wanted to do is start from a disassembled configuration and use a trigger strand in order to drive assembly of the scaffold. And to do this, we had to restructure our scaffold in order to separate the hairpins and create this blocked sequence over here. Now, upon expression of the trigger strand, this blocked sequence should open up, and then the second piece of the scaffold could come in and assemble, thus driving scaffold assembly. Again, to test this, we had all these components expressed separately with constitutive promoters. And then we had our trigger sequence under an inducible promoter. So what we did here is we had our two hairpin sequences expressed, and then we combined that expression system with an inducible expression of a fully scrambled trigger, a scrambled toehold trigger, or a regular trigger. And again, in this case, only assembly should only occur with this regular trigger. And this are the results of that experiment, where we see a fold increase in luminescence, again, calculated as this ratio of samples. And we see that for the regular trigger, we have very comparable peak in luminescence fold increase as the original system. For the scrambled toehold trigger and the fully scrambled trigger, we see some increase in luminescence. However, it is not that great. It's significantly lower than the regular trigger. One of the reasons for this is because the hairpin we designed to block the sequence is a bit unstable, and that is by design to facilitate opening of the hairpin. If somebody wanted to make a tighter binding hairpin, they could do that through designing that RNA sequence. However, that might come at the cost of lowering your overall fold luminescence, as it makes the hairpin more difficult to open. So having shown that we can kind of dynamically assemble and disassemble our system, and then turn it on or off using a trigger strand, the next question is, can we actually use this to control metabolic flux in native and non-native pathways? And to do this, first we wanted to show that we can use it in a non-native pathway. We wanted to use a two-step pathway with relatively small proteins and a product that can be quantified easily. And to use this, we chose the indole 3 acetic acid pathway, which is a plant auxin that has also been used in a previous paper that's scaffolded enzymes to demonstrate increase. So basically, it is a two-step pathway that starts from tryptophan, and you end up with indole 3 acetic acid catalyzed by these two enzymes. So to do this, what we ended up doing is basically swapping out our large bit and small bit with these IAM and IAH proteins, and creating the fusions with the two orthogonal Cas6. Now, IAM is a dimer, so basically we could expect, what we expect to see is kind of a scaffold structure that probably looks like this, although we don't really have an easy way to verify that. But what we did do is test two different levels of expressions for the proteins using TET operators. So we had a low level of expression and a high level, the high being kind of the usual induction level used for anhydrotetracycline. So post-induction, we let the cultures grow for 16 hours, collected the supernatant, and analyzed the results via HPLC. So these are the results of this experiment, with direct assembly being the original system. For the low protein expression regime, we see about a 9-fold increase in tighter product when we express the scaffold RNA. And then for the high protein, this is about 12-fold. For the high protein, we see about a 4-fold to 5-fold increase in titer. And then similarly for the trigger mediated assembly, over here, it ends up being about a 4-fold increase in product titer. And then for the high protein, it is again close to a 3 to 4-fold increase in titer. So both with the high protein and the low protein expression regimes, we see a significant increase in product titer when we express the scaffold RNA. But more importantly is, I think, the comparison between these two bars over here. So you can induce 100-fold less protein alongside your scaffold and have similar titer products to if you just induced the high protein or 100 times as much protein production in the system. So this could be a very exciting result, as you can end up basically having the cell produce a lot less protein of your non-native pathway to give you the same end product. So it makes it easier on the cell and just makes it overall a more efficient process. The next thing we wanted to check is if we can kind of redirect native metabolism for malate production. So what we did is we scaffold pyruvate dehydrogenase and isocitrate lyase. So the PDH complex with the isocitrate lyase produce two products that can be consumed downstream by malate synthase to produce malate. So because of the way these three enzymes relationship is works, scaffolding either of PDH or ICL with malate synthase would probably not work very well because you need both of these products in order to produce malate. So instead, we chose to scaffold these two. And how we envision this happening is basically when you scaffold the two proteins, you end up creating this localized region of high concentration of both acetyl-CoA and glyoxylate, which allows malate synthase to come in and catalyze the reaction between the two to produce malate. So these are the results from this expression, where again, we have our direct assembly in blue, our trigger-mediated assembly in orange, and then our negative control, which is a strain that does not have any sort of scaffolding in it. So what we see here is basically when we have our low, in this case, we tried different induction strengths of the scaffold RNA. And we see that even with a low induction level of scaffold RNA, we do get a 2.5 to 3-fold increase in malate titer for the direct assembly. And then for the trigger-mediated assembly, the difference is not as pronounced over here, but if you go to higher scaffold/trigger RNA expression, you again go to about a 2.4 to 3-fold malate titer. So it does seem that for each scaffold architecture, there is a different optimal induction strength depending on what you want. More importantly, the fact that we can just co-localize two enzymes whose products combine downstream in a pathway to increase titer also could demonstrates a novel approach for metabolic flux rewiring, where you're not only limited to kind of using enzymes that are downstream of one another, but you can now actually create more interesting combinations and even in branched pathways. So this kind of concludes the experimental work we've done so far. So to summarize, in part one, we demonstrated that our scaffold can assemble with high affinity and specificity. In parts two, we demonstrated that the assembly is, and this assembly is RNA sequence specific, and that we can also dynamically cycle through assembled and disassembled states. And then also in part two, we demonstrated that we can modularly swap out different scaffold sequences or different trigger sequences to kind of control how our system behaves. And also in part three, we demonstrated that we can control metabolic flux and also that we can easily swap out different enzymes on the scaffold to control different pathways. So it is modular on that side too. A few future directions that I see this work going to is, one thing that would be very interesting is genomic integration of components. So if we were able to basically integrate the scaffold sequences, the scaffold proteins with spike catcher, snoop catcher pairs in the chromosome, then we would only need one plasmid that can co-express our enzymes with the corresponding spike tag, snoop tag, and have everything kind of ligate inside the cell after expression. This would greatly minimize the amount of plasmids needed for the system and probably make it even more efficient. Moreover, another interesting thing could be higher order scaffolding of metabolic pathways. So by extending the scaffold and creating mutated versions of Cas6 that cannot cleave, you can end up scaffolding even more enzymes in series, where one step of the scaffold could be the one that assembles and disassembles. And this could work well for branched pathways or for bottlenecks in a pathway that you want to control how fast the throughput through that part is. Also, by using other components of that type 1 CRISPR-Cas system, we could theoretically scaffold enzymes at different ratios from one another, especially this Cas7 and Cas5 proteins. So that could be a very interesting path. Also, one of the reasons we developed this as a trigger-based assembly and disassembly is we would like to have smart native RNA, mRNA sensing scaffolds, where our scaffold is able to sense an mRNA that is produced by the bacterium as a result of a metabolite accumulation or depletion, and use that as the driver to disassemble the scaffold. And then once the cell returns to kind of the normal state, that mRNA will be downregulated again, and that'll allow you to return to your on state. Finally, this is at the end of the day, a scaffold to colocalize proteins. So I think it could have some very interesting applications in creating synthetic signaling pathways, where you can have a kinase and an activator be scaffolded together to drive some sort of downstream activation. With that said, I'd like to thank Dr. Wilfred Chen and the Chen Lab. It's been a long road, but we finally got the paper out, and the work actually culminated into something very interesting and very beautiful. I'd like to also thank my committee members, Dr. Terry Pabuchakis, Dr. Aditya Kunjapur, and Dr. Brian Bounson for all their help so far, for all their help through my thesis, and then Dr. K-Siu and EJ Kim, my lab mates who taught me a lot of what I know, and also acknowledge our funding sources. And with that, I will take any questions.
Wonderful. So it's a brilliant talk. Thank you so much. Mike?
Can I ask, I do have to go in just a
Sure.
bit, so may I ask the questions first?
Sure, please.
Again, really nice piece of work. And so the question I had is, the way you've assembled some of the switches is really clever. My question is, how stable are these constructs?
Are we talking about the scaffold constructs or the trigger constructs?
The trigger.
Likely not very stable, but it doesn't matter as of now, because essentially if you're using a synthetic trigger, it will be inducible and you can basically flood the system with it. So even if it's not stable, it's not important. But at the end of the day, it not being stable, I don't think is a very big deal, because you want to be able to, if you want to be able to switch between different assemblies, shutting down the production should be able to be enough for that trigger to go away pretty fast. I don't know if that makes sense.
Yeah, makes sense. Which is, I think halfway you had the graph with the blue line that looked like a V. I think probably illustrates some of that. So basically, the other question then is, as far as it being inducible, so the cell itself, again, you were using plasmids here, the cell constructs themselves are fairly stable, I take it?
Yes.
Okay.
So we've gone out to about 15 hours with the luminescence system, and that there was still signal there. It does kind of degrade, or it peaked at 8 hours, but it does stay pretty elevated up until that last time point we took. But yeah, and then the actual fermentations we were doing, they were 16 to 20 hour long. So, and I believe that was pretty stable. In terms of longer than that, we haven't done any studies on that. But I suspect if you wanted to keep it longer, you'd have to start diluting your culture with fresh media to keep it at a lower OD, and also to kind of help minimize. Yeah, we get a lot of growth, especially for some of the malate assays. So we want to, it gets too much and you end up having clumps of cells and it's not super efficient. So especially in test tubes.
So one last question, and then, so your last few slides, you're looking at basically secondary metabolites, correct?
Mm-hmm.
Okay. And these can tend to be toxic to the cells themselves as they accumulate. So I was wondering, was that an issue? And if so, how did you control that? Or did you just keep the concentration very, very low?
Actually, it was quite the opposite. And sorry, I have a slide somewhere
Okay.
here. So if you look at the biomass yield for glucose consumed, we actually have more yield when we induce. This yield also seems to increase with our negative control, but it's still higher for the actual scaffold systems for these secondary metabolites. In terms of the indole-3 acetic acid pathway, we definitely saw reduction in growth there, especially because indole-3 acetic acid can be toxic at high concentrations. But the reduction was not too terrible. It was maybe a 10 to 15% reduction in optical density.
Okay. Well, thank you very much.
Thank you. That's wonderful question. Actually, I wanted to ask. So I will add one more a little bit broader question. So my question is at the following. So as you may know, my PhD proposal written in a long time ago, I think 2006, is all about dynamic metabolic control. And I even coined the term glucose or metabolite valve with Kristalla Preder, my advisor at MIT. And also I started this project probably too early before people thinking about it, and could not finish it because I focused on another project, a little bit more exciting, I guess. I mean, protein scaffold project with John Duber and Jay Kiesling in UC Berkeley. So you nicely combine both of them, and congrats, I mean, impressive work. But my question is that, thank you. My question is that dynamic control is required for many living systems, but for some applications, the speed of response rate is not fast enough when we talk about or use biological systems, because, for example, gene expression takes time, sometimes many minutes to hours. Could you kind of comment on this point or challenges?
Yeah, well, the whole thought behind developing this kind of scaffold is that your only limiting factor is expression of a short RNA sequence. So it's much faster than actually having to express a different protein or doing some kind of gene silencing or something like that to downregulate. So it's only RNA expression that is the limiting factor. If you're using RNAs that are natively expressed, then it would probably be as fast as any biological process would be. I don't think there's other ways, unless we figure out a way to use small molecules as the drivers for this assembly and disassembly in a direct fashion. But yeah, the whole point of us using this is to kind of work around that. And of course, at this stage, it's still very much early on in the design and development of the scaffold. So I do believe that you can optimize the total mediated strand displacement reaction even more. And I also do think that as we learn more, we'll figure it out. But at the end of the day, this is just a toolbox in our belt as metabolic engineers. It might not be the best way to solve every non-native pathway engineering problem, but it could be very useful in cases where other approaches do not work, for example.
Of course, I mean, this is now 10 years after first paper published about dynamic control, 10 years ago by Jay Kiesling group, also Preder group. But my second question is more technical question, and I believe you are a chemical engineer, right? So you could answer that question. So, as you can imagine, there are two things you need to consider when it comes to localization, because one of them is a reaction and the other is the diffusion of the metabolite. And depending on the reaction rate and also depending on the diffusion rate of the metabolite, there are some regime that only work for localization concept. And then I'm not sure you have thought about this kind of problem because if diffusion is fast enough, or diffusion is fast enough compared to the reaction rate, your localization strategy may not work. You know what I'm saying?
Yeah, no, I completely agree with you. And again, it comes back to this isn't a solution for every problem.
Right. So,
but it will be for some. And I think the most important application for this will be in branches of either branches or very big bottleneck steps where something is very slow. So in a branched pathway, if diffusion is very fast, then by co-localizing it, you give it a much higher chance to go in the direction you want it to go versus the other one, essentially. So that is where a fast diffusion will work in our favor.
Right. So, I mean, actually, the fast diffusion, your system may not work because that's already fast diffusion, the metabolite-wise, compared to the reaction rate. The system only working when your reaction rate is not fast enough. Fast enough. In that way, you have in the regime, you kind of benefit from the localization because in that way you kind of minimize the distance between one enzyme to the other. In that way, you have fast enough time from one to the other. In that way, you have the faster conversion. So those kind of things not many people aware about. So I just wanted to bring up because when people utilize your system, I want them to aware of those kind of consideration. And then probably the simple calculation they could do is just K-cat calculation, Km calculation, and then thinking about diffusion rate. That is kind of typically, I don't remember the number right now, but on the order or something, and then you can calculate easily using simple model, and then you can predict whether the system working using your system or not, because not necessarily your system working for every single metabolic pathway.
Yeah. I mean, that was actually something I wanted to work on more, but it's surprisingly difficult to find Km and Kcat values for pathways I was looking for. You either have to generate them yourself or you have to be very lucky. So
yeah, exactly. Because nowadays not many people working on biochemistry, traditional biochemistry work, because that's tedious.
Like even the central carbon metabolism enzymes, I was trying to find some Kms and Kcats for those, and you could find maybe one or the other, and it was for the wrong variant, and it was it's quite difficult to track anything down. Yeah,
of course. I mean, that's how we did some modeling thing. I mean, when I was a graduate student, but that's even a long time ago. But I don't believe not many data accumulated over 17 years now. Oh my God, I'm getting old. But because not many people now working on this biochemistry traditional one, so that's kind of problem. So okay, so I now see my clock 11. So let me close. So thank all for joining and staying today, especially during the vacation weeks. We'll meet again next week on August 11, Thursday, the same time, the same Zoom link. We'll have Dr. Linda Chrissy from DAPA, and also she was my program manager for ONR Young Investigate Award, and so I'm super grateful for her support. But also she's the big supporter for synthetic biology, so I'm really grateful for her commitment. And also Dr. Joshua Atkins from USC. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. Thank you, and I will stop recording. Let me see. Stop recording.